Hello students, today we will learn about molecular marker aided plant breeding. So, let's begin. Plant breeders are always interested to improve the agricultural production. This is possible to produce new improved plant varieties with the desired characteristics. An important task of plant breeders is correct identification and continued maintenance to the plants with improved characters. There are different types of markers used for this purpose. The basic requirement of a marker is that it must be polymorphic. That is, it must exist in different forms. The polymorphism can be detected by three different types of markers. First is morphological markers, biochemical markers, and third is molecular markers. First, we will discuss about morphological markers. The morphological or phenotype markers represent the quantitative traits that can be visually detected. Example: altered leaf morphology, albinism, dwarfism, etc. Morphological markers may be dominant or recessive. These markers are not widely used to detect polymorphism these days due to some limitations. Let's know about the limitations. Morphological markers influenced by the environment and thus may not represent the desired genetic variation. Majority of the visible markers may not be required in the plant improvement programs. Now let's know about biochemical markers. The proteins produced by gene expression are also used as markers in plant breeding programs. The most commonly used are isoenzymes, the different molecular forms of the same enzyme. The isoenzymes can be separated by electrophoresis and identified. However, the polymorphism of the isoenzymes is rather poor, hence they are not widely used as markers. Now let's know about molecular markers. A molecular marker is a DNA sequence in the genome which can be located and identified. As a result, the genetic alterations, that is mutations, insertions and deletions, the base composition of a at a particular location of the genome may be different in different plants. These differences, collectively called as polymorphism, can be mapped and identified. Plant breeders always prefer to detect the gene as a molecular marker. Although this is not always possible, the alternative is to have markers which are closely associated with genes and inherited together. The molecular markers are highly reliable and advantageous in plant breeding programs. The molecular markers provide a true representation of the genetic makeup at the DNA level. They are considered consistent and not affected by environmental factors. Molecular markers can be detected much before development of plant occurs. A large number of markers can be generated as per the needs. Now let's know about the basic principles of molecular marker detection. Let us assume that there are two plants of the same species, one with disease sensitivity and the other with disease resistant. If there is DNA marker that can identify these two alleles, then the genome can be extracted, digested by restriction enzymes and separated by gel electrophoresis. The DNA fragments can be detected by their separation. For instance, the disease-resistant plant have a shorter DNA fragments while the disease-sensitive plants may have a longer DNA fragment. Molecular markers are of two types. First is based on the nucleic acid, that is DNA hybridization or non-PCR based approaches, and second is based on PCR amplification, PCR based approaches. Markers were based on DNA hybridization. Let's know about the markers based on DNA hybridization. The DNA pieces can be cloned and allowed to hybridize with the genomic DNA, which can be detected. Marker based DNA hybridization is widely used. The major limitation of this approach is that It requires large quantities of DNA and the use of radioactivity. Now let's know about the RFLP, restriction fragment length polymorphism. RFLP was the very first technology employed for the detection of polymorphism based on the DNA sequence differences. RFLP is mainly based on the altered restriction enzyme sites as a result of mutations and recombinations of genomic DNA. The procedure basically involves the isolation of genomic DNA, its digestion by restriction enzyme separation by electrophoresis, and finally hybridization by incubation with cloned and labeled probes. Based on the presence of restriction sites, DNA fragments of different lengths can be generated by using different restriction enzymes. Two DNA molecules from two plants, suppose A and B, are taken. In plant a mutation has occurred leading to the loss of restriction site that can be digested by EcoR1. The result is that when the DNA molecules are digested by the enzyme Hnd3, there is no difference in the DNA fragments separated. However, with the enzyme EcoR1, plant A DNA molecules is not digested while plant B DNA molecule is digested. This results in a polymorphic pattern of separation. Now let's know about the markers based on PCR amplification. Polymerase chain reaction is a novel technique for the amplification of selected regions of DNA. The advantage with PCR is that even a minute quantity of DNA can be amplified. Thus, PCR-based molecular markers require only a small quantity of DNA to start with. PCR-based markers may be divided into two types. First is locus non-specific markers. Second is locus specific markers. Let's know about the locus non-specific marker. That's example. Let's see. Example, random amplified polymorphic DNA, that is RAPD, and amplified fragment length polymorphism, that is AFLP. And locus specific markers, example, simple sequence repeats and single nucleotide polymorphism, SNP. Now, let's know about random amplified polymorphic DNA markers. In short, RAPD. RAPD is a molecular marker based on PCR amplification. The DNA isolation and the DNA isolated from the genome is denatured. The template molecules are annealed with primers and amplified by PCR. Single short oligonucleotide primers, usually 10 base pair, can be arbitrarily selected and used for the amplification DNA segments of the genome. The amplified products are separated on electrophoresis as identified. Based on the nucleotide alterations in the genome, the polymorphism of amplified DNA sequences differ, which can be identified as bends on gel electrophoresis. Genomic DNA from two different plants often results in different amplification patterns, that is RAPDs. This is based on the fact that a particular fragment of DNA may be generated from one individual and not from the others. This represents polymorphism and can be used as a molecular marker of a particular species. Now let's know about AFLP, amplified fragment length polymorphism. AFLP is a novel technique involving a combination of RFLP and RAPD. AFLP is based on the principle of generation of DNA fragments using restriction enzymes and oligonucleotide adapters and their amplification by PCR. Thus, this technique combines the usefulness of restriction, digestion, and PCR. The DNA of the genome is extracted. It is subjected to restriction digestion by two enzymes. The cut ends on both sides are then ligated to no sequence of oligonucleotides. Now come to PCR. PCR is now performed for the pre-selection of a fragment of DNA which has a single specific nucleotide. By this approach of pre-selective amplification, the pool of fragments can be reduced from an original mixture. In the second round of amplification by PCR, 3 nucleotide sequences are amplified. This further reduces the pool of DNA fragments to a manageable level, that is more than 100. Autoradiography can be performed for the detection of DNA fragments. Use of radio-labeled primer and fluorescently labeled fragments quickens FLPs. FLP analysis is tedious and requires the involvement of skilled technical personnel. Hence, some people are not in favor of this technique. In recent years, commercial kits are made available for FLP analysis. FLP is very sensitive and reproducible. It does not require prior knowledge of sequence information by AFLP. A large number of polymorphic bands B can be produced and detected. Now let's know about STS, that is sequence tagged sites. Sequence tagged sites represent unique simple copy segments of genomes whose DNA sequences are known and which can be amplified by using PCR. STS markers are based on the polymorphism of simple nucleotides repeats, that is GAN, GTN, CAAN, etc. on the genome. STS have been recently developed in plants when the STS loci contain simple sequence length polymorphism SSLPS. They have high valuable as molecular marker. STS loci have been analyzed and studied in number of plant species. Now let's know about microsatellites. Microsatellites are the tandemly repeated multicopies of mono, di, tri and tetra nucleotide motifs. In some instances, the flanking sequence of the repeat sequences may be unique. Primers can be designed for such flanking sequences to detect the sequence tagged microsatellites. This can be done by PCR. Now let's know about sequence characterized amplified regions. SCARs. SCARs are the modified forms of STS markers. They are developed by PCR primer that are made for the ends of RAPD fragment. The STS converted RAPD markers are sometimes referred to as SCARs. SCARs are useful for the rapid development of STS marker. Now let's know about molecular marker assisted selection. Selection of desired traits and improvement of crops has been a part of the conventional breeding programs. This is predominantly based on the identification of phenotypes. It is now an accepted fact that the phenotypes do not necessarily represent the genotypes. Many a times the environment may mark the genotype. Thus, the plant's genetic potential is not truly reflected in the phenotypic expression for various regions. Now, the molecular marker assisted section is based on the identification of DNA markers that link or represent to pathogens and insects, tolerance to abiotic stresses and various other qualitative and quantitative stresses and various other qualitative and quantitative traits. The advantage with molecular markers is that a plant breeder can select a suitable marker for the desired trait which can be detected well in advance. Accordingly, breeding programs can be planned. There are some major requirements for the molecular markers selection in plant breeding. Let's know about the major requirements. First point is the marker should be closely linked with the desired trait. Second point is the marker screening methods must be efficient, reproducible and easy to carry out. Third point is the analysis should be economical. Now let's know about molecular breeding. With rapid progress in molecular biology and genetic engineering, there is now a possibility of improving the croplands with respect to yield and quality. The term molecular breeding is frequently used to represent the breeding method that are coupled with genetic engineering techniques. Improved agriculture to meet the food demands of the world is a high priority area. For several years, the conventional plant breeding programs have certainly helped to improve grain yield and cereal production. The development of dwarf and semi-dwarf varieties of rice and wheat have been responsible for the green revolution, which has helped to feed millions of poverty-stricken people around the world. Many developments in the agriculture front are expected in the coming years as a result of molecular breeding. Now let's know about the linkage analysis. Linkage analysis basically deals with studies to correlate the link between the molecular marker and desired trait. This is an important aspect of the molecular breeding programs. Linkage analysis has to be carried out among the populations of several generations to establish the appropriate linkage. In the earlier years, linkage analysis was carried out by use of isoenzymes and the associated polymorphisms. Molecular markers are now being used. The techniques employed for this purpose have been already described till now. Now, let's know about quantitative trait locking. There are many characteristics controlled by several genes in a complex manner. Some good examples are growth growth habit, yield, adaptability to environment, and disease resistance. These are referred to as quantitative traits. The location on the chromosome for these genes are regarded as quantitative trait loci. The major problem the plant breeder faces is to is how to improve the complex character controlled by many genes. It is not an easy job to manipulate multiple genes in genetic engineering. Therefore, it is very difficult and time consuming process. For instance, development of golden rice involving the insertion of just 3 genes took about 7 years. Now let's know about arid and semi-arid plant biotechnology. The term arid zone is used to refer to harsh environmental conditions with extreme heat and cold. The fields have limited water and minerals. It is different task to grow plants and achieve good crop yield in arid zones. Semi-arid regions are characterized by unpredictable weather, or inconsistent rainfall, long dry seasons, and poor nutrients in the soil. Most parts of India and many other developing countries have semi-arid regions. Crops like sorghum, millet, groundnut, and cowpea are mostly grown in semi-arid tropics. Besides unpredictable weather, biotic and abiotic stresses contribute to crop loss in these areas. The biotechnological approaches for the breeding programs in the semi-arid regions should cover the cover some areas. Let's know about the areas. What the areas should be. First is development of crops that are tolerant to drought and salinity. Second is improvement to withstand various abiotic and biotic stresses. Third point is micropropagation techniques to spread economically important plants which can withstand harsh environmental conditions. Next is some success has been achieved in improving sorghum, millet and legume crops that are grown in semi-arid regions. Genetic transformation in sorghum was possible by using microprojectile method. Now let's know about the greenhouse and green home technology. What is the difference we'll study? Greenhouse literally means the building made made up made up of glass to grow plants, or we can tell also transparent like polythene we can use also. Greenhouses are required to grow regenerated plants for further propagation and for growing plants to maturity. Greenhouses are the intermediary stages involving the transitional step between the plant cultures and plant fields. The purpose of greenhouses is to acclimatize and test the plants before they are released into the natural environment. The plants are grown in greenhouse to develop adequate root systems and leaves so as to withstand the field environment. The greenhouses are normally equipped with cooling systems to control temperature. Greenhouses have chambered fitted with artificial lights. It is possible to subject the plants to different lighting profiles. In recent years, many improvements have been made in the development of more suitable greenhouses. These include the parameters such as soil and humidity. The major limitation of greenhouse technology is an increase in carbon dioxide production that in turn increases temperature. Some approaches are available to control temperature. Green home technology. Let's know about green home technology. What is green home technology? Green home technology is a recent development. In this case, temperature is controlled by using minimum energy. Now completed this topic. Any questions regarding this? You can ask me in the comment box and subscribe my channel to know more biotechnological topics. Thank you.